This is Where We Live from Connecticut Public Radio. I'm Catherine Shen. One of the things that makes Where We Live so special is our proximity to Long Island Sound. This estuary and its 600 miles of coastline is home to nearly 200 fish species, more than 1,000 invertebrates, and dozens of migratory birds. This is according to Audubon, Connecticut. And this morning, we're kicking off NautiWeek here at Connecticut Public, where we're going to spend a week to celebrate all things nautical. And here on Where We Live, we're focused on this unique ecosystem. Stay tuned for deep dives on terrapin turtles, sturgeon, jellies, and oysters just off our coast. And today, we're diving for coral.
They got a lot of great colonies.
There's a bunch on this one. Wow, good score.
Tropical coral populations are on a steep and troubling decline. Coral in the Florida Keys is experiencing a mass bleaching event due to hot tub level water temperatures. Here in Connecticut, researchers hope one native species of coral can help its more vulnerable relatives. Coming up, we'll hear from researchers with the Temperate Coral Research Group. But first, here to discuss how climate change is showing up in Long Island Sound is Bill Lucey, who's a soundkeeper with Save the Sound. Thank you so much, Bill, for joining us this morning.
Pleasure to be here.
And have you observed any changes to the shoreline where we live? You can join the conversation, 888-720-9677, or find us on Facebook and Twitter at Where We Live. So, Bill, a lot of build-up to this first question here. I want to ask you the big picture and long-term. What are your concerns for Long Island Sound when we're talking about climate change?
Well, I would have to say the primary concern, obviously, is water temperature. If you look at the data over the last few decades, there's a steady, small rise. I was looking at a report by some colleagues at Project O, and they're looking at about 0.45 degrees Celsius over the last 10 years. So, looking at 0.8 of a degree Fahrenheit for those who like to think in Fahrenheit. So each year on average, we're creeping up. And the reason that worries me so much, just sticking with the physical aspect of it, is that Long Island Sound's had a history of hypoxia. Back in 1994, when it peaked, hypoxia is an area where there's zero oxygen. So we had 212 square miles of hypoxic conditions back in 1994. And that's when, if you remember the stories, lobsters were dying in the pound and there was bunker kills every day. It was terrible. So there was a big action that happened called the Long Island Sound Nitrogen Action Plan to try and reduce the nutrients. And starting about 17, 18 years ago, they put limits on all the wastewater treatment plants because they all have clean water permits under the law and how much they're allowed to discharge. And over the course of the past 17, 18 years, we've really seen an effect. So in 2022, the hypoxic area was down to 53 square miles. So you remember back in 2012 or 1994 was 212, and that lasted for about 70 days. It's decreased quite a bit. Now there's fluctuations, and these are averages. But if you look at just the data from when they initiated the nitrogen action plan till now, there's a significant drop. Now, the problem is we're making great gains on our effluent discharge, but we have increase in population, non-point source runoff, and things are getting warmer. So warm water holds less oxygen. So we might start seeing some increases in hypoxia unless we get a handle on our climate activities.
So a lot of things going on, really, and we see so many headlines related to this recently. We also spoke with Justin Davis with the State Department of Energy and Environmental Protection's Marine Fisheries Division late last week, who also spoke about unpredictability in the Sound. Let's take a quick listen.
One thing that we've seen with what's going on with climate in our region, and boy, this summer has been a great example of it, is that the weather has just gotten more volatile and more unpredictable, right? So if you go out in Long Island Sound right now, the water is chocolate brown. We've had a huge influx of fresh water because of a really unseasonable flood in the Connecticut River. There's a ton of suspended sediment in the water. I've heard from fishermen that it has driven fish out of areas where they would normally be at this time of year, which has impacted fishing activity. So things like this, where climate is producing just greater volatility in the environment, leading to unpredictability and sort of where important fish species might be over time, might create impacts in the nearshore environment with tons of sediment coming in from big rainfalls washing lots of nutrients into the water off the surface of the land that could cause algal blooms that leads to, the low oxygen hypoxia conditions you talk about. So I think that's certainly one thing we're keeping an eye on and acknowledging, is that just the environment's getting less predictable. Not that it's ever been really predictable to begin with, but it's getting even more unpredictable going forward.
So I'm hearing some echoing there with hypoxia. Bill, what's your response with what Justin has to say, especially when we're thinking about the impact on the local ecosystem here?
Yeah, well, stochastic events, these unforeseen events create things like out of sync algal blooms. We also see, with sewage discharges and other nutrient sources, algal blooms happening. And one of the long-term trends of that is that we're seeing a shift of our basic zooplankton community. So back in the 50s, there was a couple species of copepod that were big and juicy, really tasty to eat. And as hypoxia became more of a problem, as populations increased, you had a loss of those species. Two medium-sized species took their place, but now we're seeing a decline in those, and a smaller species is showing up. And the reason that's important is that copepods are the link between the sunlight and plankton, which takes that sun energy and the nutrients and makes it into salad, let's say. Then the zooplankton eats that, and the zooplankton in turn feeds all the larval fish and small forage fish species. So if a fish has evolved to eat a certain species at a certain time, and that starts to change, it can have effects on both the larval species and really on forage fish, which support the whole food chain. And I was really surprised. Most of my time was spent in Alaska, and we get our spring blooms in April, May, but there are blooms that'll happen in January in Long Island Sound. And so the bloom timings can really change what's going on with the copepod. They'll smother some of the silica, other things that diatoms need to take up, which feed these copepods. And it's stuff you just don't see. So Justin's absolutely right. There's small changes that we can't see all of a sudden reflect themselves in how'd you do fishing today, or how'd you do this season versus last season. It's the great unknown.
Right, and those small changes seem to lead to bigger changes, as we're having this conversation today. And like we just heard from Justin Davis with DEEP's Fisheries Division, he also spoke to regulatory delays where it concerned changes to fish populations due to climate change. Let's take another listen.
A sort of interesting challenge we've had, too, with fish species that used to live down south moving up here into Long Island Sound, is that our regulatory structures, sort of the rulemaking process, has not been as quick to catch up to what's happening out there in the environment. One of our missions is about sort of setting the rules around fishing, how many fish people can take out of Long Island Sound. We don't get to make those decisions in-house unilaterally ourselves in Connecticut. We do it through what's called the Interstate Fisheries Management Process. It's a complicated way of saying that we have to make those decisions in cooperation with all the other Atlantic coastal states, because those fish that are really valuable in Long Island Sound, a lot of them move up and down the coast. And so we all have to work together on the coast to set the fishing rules. Problem is that because these fish historically live down south, most of the what we call the quota, or sort of the annual allowable take, was held by southern states. As those fish have moved up here, the management structure hasn't responded as quickly. So even though the fish have become really abundant here in Long Island Sound, our fishing community hasn't been able to take full advantage because we just don't have the access we probably should with the abundance of the fish there.
So sounds like there's so many moving parts to fishing here, Bill. You're also involved in a lot of regulatory and legislative conversations. What's your perspective on this? Has change been happening? What's going on there?
Yeah, well, Justin is absolutely correct. The fishermen have been taking the brunt of climate change with no regulation, right? The lobster fishery has crashed. The winter flounder have really gone downhill. So those opportunities aren't there anymore. The opportunities that are showing up, as Justin said, are regulated farther south. So let's take black sea bass. There's a lot of black sea bass. Any of the fishermen out there, whether they're sport anglers or commercial, are going to have seen this dramatic rise in black sea bass over their careers. And so there's an opportunity to replace some of the lost income for these harvesters. But as Justin mentioned, the regulations don't keep up. The climate change happens. Those opportunities are lost. So we really need, and I know some of the Connecticut delegation has been trying to push this along. We need to create more opportunities for our local fleet.
And as we continue to have these conversations, and I don't foresee us stopping really anytime soon, do you have any bigger picture thoughts on how regulation can better keep up with climate change?
You don't want to do things too fast. You have to be careful about using legislative policy to manage natural resources, but sometimes that's a way to make things move more quickly. We just need to be actively engaged as a state, and I think we have really good fisheries leadership in the state of Connecticut right now, and we just need to keep pushing for more equitable quota shares.
And so you mentioned sea salad earlier. We know Save the Sound is also working on eelgrass restoration in the Sound. Can you tell us what eelgrass is and the important role that it plays in this ecosystem?
Sure. Eelgrass is one of the unsung heroes of the near coastal areas. It's submerged aquatic vegetation. We call them SAVs. Used to be very abundant in Long Island Sound. It's a great place for larval fish and crabs and all kinds of eggs and things to settle. There's a symbiotic relationship with shellfish, especially scallops and other bivalves, which fertilize the seagrass, and the seagrass catches all the nekton, all the good stuff for those clams to filter. But back in the 30s, there was a large die-off from a wasting disease. And it wasn't until the 50s there was a resurgence. But in the 60s, it started to drop off. We had a lot of sewage inputs, rising population, and right now we're to the point where eelgrass is pretty much restricted to the Eastern Sound. So we've been looking at trying different ways to go to areas that may not have seeds. The founding population or nearby populations they're too far away to restore an area. So if the water quality has gotten to a point where maybe we could try eelgrass again, it needs a helping hand. So what we've been doing is collecting seeds, gluing them on clams, and dropping them overboard and seeing how they grow. And we're shooting about 50%. It's worked in some areas, hasn't worked in others. Eelgrass rest. There's some really good researchers in the Sound that are focused on this right now.
And so, we've been talking a lot about movement, and there are some bright spots of research that's happening off our coast with the Northern Star Coral, another hero, I believe. And we only got about 2 minutes left here, but I would love to hear about your hopes as far as the role of Long Island Sound can play in climate resilience.
Yeah, well, just taking eelgrass, it's one of the ecosystem services is it sequesters carbon. And I know Stony Brook, Brad Peterson at Stony Brook University has been working on how quickly they can catch and store carbon. And UConn, Dr. Jamie Baudry has been looking at how iron can mitigate some the bad hydrogen sulfide that comes out of our nasty sediments that have too many nutrients to spur growth. And then Cornell Cooperative Extension has been really piloting several different techniques to bring it back. So I think we're in an area that if we can, as a region and a couple of states working on this, come up with some viable restoration strategies that can be exported to other parts, that can help everybody, because eelgrass is an issue globally. We also had the Long Island Sound study, which has finally been funded to its fullest extent. And we have a lot of good researchers along the Sound, and we have a lot of people trying to put that research into action through policy and restoration practices. So, things are tough right now, but I think we have the tools to really turn it around and set things on a healthy path.
Well, that sounds like a plan. And here's three huzzahs for the eelgrass. Long Island Soundkeeper Bill Lucey, thank you so much for being with us this morning.
For sure. Thank you very much for having me.
Coming up, we hear from researchers focused on one species of Connecticut coral and about their hopes for helping its tropical relatives that are more threatened by warming waters. You can also join the conversation, 888-720-9677. That's 888-720-WNPR, or find us on Facebook and Twitter at Where We Live. This is Where We Live from Connecticut Public Radio. I'm Catherine Shen. There's a coalition of scientists along Long Island Sound and Narragansett Bay who are focused on the native species of northern star coral, or Ostrangia poculata. The Temperate Coral Research Group hopes their research could lend a hand, or tentacle, to tropical species more threatened by warming. Joining us now is Sean Grace. He's a marine ecologist and biology professor at Southern Connecticut State University, and he also co-directs Southern's Worth Center for Coastal and Marine Studies. Thank you so much, Sean, for being with us today.
Oh, thank you so much for having me, Catherine. Good morning.
And do you have a passion for coral? Are you a fan of coral? You can join the conversation, 888-720-9677, or leave us a comment on Facebook and Twitter at Where We Live. So, Sean, can you explain what makes northern star coral such a prime candidate for study, particularly when we're talking about climate change, and besides the fact that it has a really cool name?
Oh, I love its name. Well, I think first because it has a really large geographic range, and we are at its northern limit right now. So it's southern New England's only scleractinian coral. And by southern New England, I mean Massachusetts, Rhode Island, Connecticut, all the way through probably northern or parts of North Carolina. And then there are other corals that kind of mix in after that. So this is truly New England's only coral. And I like it because it's got some unique characteristics that can give us some insight into potentially, what might be happening with corals around the world. So it has a unique symbiosis, which is a very dynamic symbiosis, meaning, corals get their color essentially from symbiosis with an algae or zooxanthellae that lives within them. And that zooxanthellae photosynthesize, and it provides the coral some food, basically. And the coral provides the zooxanthellae a home. So, and the coral that lives locally, Ostrangia, is strange in that if you look at that symbiosis, it's not always as colorful as it could be. It's not always white or where it doesn't have the zooxanthellae. So it's just very unique, and it's very different from most corals around the world.
I just want to let our listeners know that you can find images of northern star coral on our website at ctpublic.org/where we live. And Sean, you mentioned coral around the world. And to be honest, when I think coral, I don't think about New England. Can you talk to us about whether or not we might call bleaching? That's something that we've been seeing in the headlines recently. Can you walk us through how we would define that and how that's a little different here in temperate waters?
Yeah, sure. Well, if we bring ourselves to a tropical reef, I think one of the striking images that we see are just how colorful corals are, right? And they're beautiful. Right, so they get that color again from that zooxanthellae that lives within their tissues. So what bleaching is basically a whitening of the coral, and that results from a loss of that zooxanthellae or a decrease in the zooxanthellae's photosynthetic pigment. So what we're seeing in the tropics, specifically this summer, and in the past too, but it's really happening now, is a lot of bleaching events taking place. And it's a little different when we look at Ostrangia, because Ostrangia can lose its zooxanthellae and gain zooxanthellae at different times during the year. And it does just fine. So it doesn't seem to be as affected by, having no zooxanthellae like the tropical corals do.
And you've also been diving off the coast of New England for years. What sort of
Yes.
habitat changes have you observed?
Oh, geez, the last 35 years, just like local habitat changes. The kelp populations used to dominate the subtidal from the low intertidal down to maybe 7-8 meters depth. And the kelp is now gone. We don't really find kelp. We find small remnants of their populations, but nothing like it used to be. And it's been replaced with this kind of like turf-like algae, and we call it turf because it's more like replacing the tree with a bunch of bushes, right? So they're closer to the substrate, and they trap a lot of sediment, and they can smother organisms that live on those sediments, including corals. So we've seen that change and take place over the last 15 years.
And all of this, of course, impacts corals' habitat as well. Have you seen any changes specific to the northern star coral?
Well, just recently, I think this past winter, the water temperatures didn't get to the point where the coral would experience what it normally does in the winter, which is a phenomenon known as quiescence or dormancy. So these corals actually go dormant in the winter. So when temperatures get below around 4 degrees Celsius, they kind of pull in their tentacles, they puff out like their oral plates, and they just kind of remain dormant until things warm up in the spring. And then their tentacles come back out and they start feeding again. And this winter, in January, February, and March, we never really hit the temperatures that initiate quiescence in the coral. And, we observed the corals being open and feeding all 3 months. And we brought some in and actually, just to check what they were feeding on. And yeah, they were actively feeding throughout the entire winter, which for me, this is the first winter that I've experienced this. Usually the corals go dormant.
And this is so special to this coast. And I know you touched on it a little bit earlier, but what inspired you to study the northern star coral specifically?
Well, I used to do studies in the tropics. And I was looking for PhD programs, and I knew that Dr. Michael Pilson, who was at URI's Graduate School of Oceanography, had multiple students studying this coral. But this was like maybe 10 years before I went to URI to do my graduate work. And, I had worked for a coral biologist for years, and I mentioned, I think I'm going to go to Rhode Island. And it was funny. Initially, they were like, you might find 5 or 6 corals here and there. You might see a few, but you're not going to have enough to actually do a whole study on. And it took me a little while, maybe 1 or 2 dives until I actually realized that I was surrounded by hundreds of thousands of colonies of corals. And there was plenty to do research on. So for me, I'm local. I grew up in Massachusetts, and I spent a lot of time on the beaches in Rhode Island. So it was just a very natural progression of, let's study what's in my backyard.
Well, I love that too, because you went from tropics to the New England coast, not the most, straightforward transition.
Yeah, definitely not. Definitely not. I've heard about it for years. Why would you go to Rhode Island to study corals?
Well, we're having a very important conversation because of that. So I want to thank you for doing that. And perfect transition, because joining us now are 2 Rhode Island-based members of the Temperate Coral Research Group. We have Koty Sharp, who's Associate Professor of Biology, Marine Biology, and Environmental Science at Roger Williams University, and Amy April, who's an Associate Scientist at Woods Hole Oceanographic Institution. Thank you so much, Koty and Amy, for joining us this morning.
Thanks for having us.
Thanks for having us.
And Amy, I want to start with you. You studied coral in tropical and temperate settings. What are your concerns, especially hearing about the recent bleaching in the Florida Keys?
Yeah, thanks for asking about that. It's really catastrophic what's happening now in Florida. And so I'm deeply concerned, as well as researchers around the world. We just haven't seen this scale of bleaching and mortality in the Caribbean ever. And the water temperatures are basically just getting so warm that they're causing, as Sean described, that disassociation between algae and the coral. And then in some cases, the water is so warm that we're not even seeing bleaching, that there's just mortality of the coral itself are dying. And one of the reasons that this is so troubling for coral and Florida in particular is because they've just spent the last kind of 7 or 8 years experiencing an unprecedented disease event. So they lost about 50 million coral colonies just from the stony coral tissue loss disease. That was starting to dissipate. They were just starting to get their hands on some restoration activities in that area, and now we're seeing many of the corals dying there now because of the warming.
Well, and honestly, in preparation for this conversation, when I saw the news, my heart dropped because I had seen that they were sort of coming back in numbers, and just suddenly they're all bleached. It's as you said, it's very troubling. And I want to ask too, how would you describe the links between the northern star coral and its tropical relatives that we're talking about now?
Astrangia is the incredibly resilient coral that I think tropical corals are going to be striving to become. So there's clear links. Astrangia, like the tropical corals, also builds a calcium carbonate skeleton. It builds habitat for other organisms. Both Astrangia and the tropical corals host a variety of symbionts, so organisms that are smaller that live within their tissues. So the zooxanthellae that Sean described, they can live with or without them. So they figured out, hey, sometimes I need these and sometimes I'll just feed. And we're still not quite sure, how they make that decision. Astrangia, as well as tropical corals, also have bacteria and bacterial relatives that live within their tissues and on their surface, and that helps them with nutrient cycling as well as immunity. And they're all beautiful too. I think that's inspiring as well. Even when I look at an Astrangia in the water, it's just inspiring to see this organism that has been able to do so much with how it lives and how it has evolved symbiosis to thrive in these environments.
Well, I love seeing the photos of them and just very inspired that they can do so much. And on top of that, too, Koty, what would you add about the connections to tropical coral here? And what are your hopes for studying northern star coral?
Yes, I think that really the hope lies in the fact that there are so many similarities between Astrangia and our tropical corals, as Amy mentioned. And then also those really unique characteristics that Sean highlighted. So Astrangia hosts a diverse microbiome on its surface and inside of its tissues. And it's a fairly complex microbiome, but it's actually also very similar to the microbiomes that we see in tropical corals. We know that microbiomes are very important for the health of their animal hosts. And the work that we've been doing is trying to understand how Astrangia microbiomes contribute to the health of their hosts, how they respond to these environmental fluctuations, and how they might be involved in responding to the extreme temperatures of our hot summers as well as our cold winters here in New England. We joke that Astrangia is hardy like a New Englander. It can handle these really strong seasonal fluctuations. And so really one of our main focuses with Astrangia is sort of focusing in on that unique characteristic of being able to handle these large thermal changes and trying to understand the mechanisms by which they can accomplish that.
And you mentioned the microbiome. Can you tell us what exactly that makeup is for a coral? Because when I think microbiome, I'm thinking guts, my own gut, actually.
Sure, and that's a really great parallel to make, actually. So we know that we've seen a lot in the news about gut microbiomes and in humans, right, and how they're linked to health and to disease. And the same is really true for nearly all animals and all ecosystems. The microbiome, which is a community of many different types of bacteria and fungi, it consists of microbes. We focus on the really enormous number of beneficial microbes that are on the corals. And we know that in corals in particular, microbes that live on them and in them are responsible for helping out with nutrient cycling. They handle waste. They can provide immunity, as Amy said, and protect the corals against pathogens that potentially are in the water column. So they're really, really critical for supporting the health of the animal host of the corals.
You've been listening to Koty Sharp, Amy April, and Sean Grace with the Temporal Coral Research Group, and we'll be continuing this conversation after a break. Coral fans, give us a call, 888-720-9677. That's 888-720-WNPR, or find us on Facebook and Twitter at Where We Live. This is Where We Live from Connecticut Public Radio. I'm Catherine Shen. We're back with members of the Temporal Coral Research Group, Sean Grace, Koty Sharp, and Amy April. And just a reminder for our listeners, if you're a coral fan, you can also join the conversation, 888-720-9677, or leave us a comment on Facebook and Twitter at Where We Live. So, Koty, I want to jump back at this with you. We were lucky enough to intercept a collection process with you and some of your students. Can you describe that process for us?
Sure. Well, we know that based on research that was done nearly 40 years ago, we know that Astrangia produces gametes, so they produce eggs and sperm that they release into the water to meet each other in the seawater to fertilize and produce new young corals. And this is the way that they sexually reproduce in the wild. We know based on research that was done about 40 years ago that the seasonality of this is that the eggs and sperm are released in late July, August, September. And so during this time of the year, we get together and we collect corals. Because those gametes are so small, we bring those corals back into the lab and we spawn them in the lab. So we've developed techniques over the past 7-8 years now with a large group of collaborators from multiple institutions that allow us to spawn these and get eggs and sperm in the laboratory and then fertilize them in the laboratory and raise them in captivity. We call this event that we've been working on Spawn Fest. And because like all marine scientists, we like to give things snazzy names. And at Spawn Fest every year, we basically spend a week or two really dedicated to focusing just on getting a lot of spawn in the lab and learning as much as we possibly can during this small window of time about the early life stages of this coral.
Well, I think you're in the right place. We love a snazzy name as we're talking about Spawn Fest as we debut NautiWeek here on Connecticut Public, where we celebrate all things nautical. So I think we're on the same page here. And I want to ask too, is the hope to confirm that these coral went dormant this year, and that's important, right?
Well, I think Sean's observations this year about the coral not reaching that dormancy are, number one, unfortunately not very surprising because, like he said, we didn't hit those lower temperatures and sustain at those lower temperatures that we usually do, those low temperatures that trigger the dormancy in the coral. The goal of SpawnFest is not so much, we didn't initiate these work trips to monitor for changes in the environment. To be totally honest, we did this because we're working on trying to develop this as a useful experimental animal for people, researchers in all kinds of different fields, to learn as much as we can about fundamental coral biology. So like so many different research topics these days, we accidentally have found another way to monitor, a very sensitive way to detect for changes in our local animals. And so as we continue to follow the spawning rhythms and reproductive output of corals in the face of climate change, that may provide us a window into how the corals are changing, how animals in our ecosystems are changing. But it wasn't, I would say, the original intent for starting monitoring this.
And Koty, this is part of what makes northern star coral such ideal candidates for study, that we can spawn enough to prove something like this?
Yes. So the spawning techniques that we use to study Astrangia and its early life stages are all modified and adapted from methods that originally were developed for studying spawning and reproduction in tropical corals. The methods that have been so finely tuned now by tropical coral researchers and have been used in land-based nurseries and restoration efforts around the globe. We've modified those for Astrangia, and we're hoping to be able to use those to answer some really fundamental questions about Astrangia's biology.
And Sean, you had shared with us a little bit about the process earlier and your philosophy behind this study. How does what Koty just mentioned now, reflect on what you've observed?
Well, I'm really excited about what Koty's studying, and when SpawnFest happens every year, especially what the data is going to demonstrate this year. Just because it'll be the first time that we'll have a spawning event after a year when the coral didn't quiesce or go dormant. And to me, even though it's like the first bit of data, it could represent like the small changes that Bill mentioned earlier, right? So these small little changes that happen every single year, that over years, maybe 10 years from now, we see a big change taking place. So, for me, Koty's research is very exciting because it's just going to be data points, after the first time that we didn't notice dormancy in this coral. So I'm very excited about that.
And Amy, what else would you add to everything that we were just talking about now?
Yeah, in addition to, temperature, which we've been talking about with this coral, I think it's important to understand that, these corals are also in an ecosystem that's changing, these northern sea star corals as well as the tropical corals. But, as Bill talked about, there might be other things like we think it's temperature that's directly related to the hypoxia, to the warming, but we know that hypoxia is occurring more and more in some of these regions of the ocean. So that will be important for us to monitor and to understand. I'm also very interested in what the warming is doing to the seawater microbes as well. This is something people aren't really talking about yet, but temperature is a major growth factor for bacteria. And so it will cause some bacteria to grow faster and will cause some of them to express genes that may actually be harmful in some cases. It can make bacteria turn into pathogens. And so, as these corals are in a warming ocean, there's other what we call synergistic impacts, so other impacts that are probably acting on their biology. And so this is one of the reasons it's so important to have a system like Astrangia where we can bring it into the lab. We can spawn it like Koty's doing. We can produce more corals, and we can do experiments to understand what's happening. And what we use, systems like Astrangia to do is to make relationships and wider statements about what's likely happening in the biology of other species and other, including corals and beyond, because you can't bring every organism into the lab and do these tests appropriately. But we're starting to be able to do that with Astrangia, and that's really exciting.
And Sean, with what Amy is saying, even resilient northern star coral may be dealing with its own impacts of climate change beyond even dormancy. Do you know what sort of impacts you're seeing already, or are you seeing those impacts?
Well, I know I mentioned that we're seeing a lot of turf algae now in the shallow areas, and it's kind of expanding into, typical coral areas, right? So, on the horizontal substrates and vertical substrates. And, we know that, marine heat waves have caused this decrease in kelp, this increase in turf. And with this turf, it's just smothering a lot of corals. I have a student right now examining that at Fort Wetherall in Rhode Island, and yeah, it's amazing how many dead skeletons we're finding underneath the turf. Whereas just immediately adjacent areas that no turf's present, the corals seem to be just fine there. So yeah, just a result of marine heat waves and what's happening to the substrates underwater.
And again, for our listeners, you can find images of that collection process on that turf on our website at ctpublic.org/wherewelive. And Koty, the northern star corals forms in relatively small colonies, and they're not reef building. Can you tell us why is that?
Well, we think that it is probably linked to these seasonal fluctuations. So researchers over the years have seen a lot of growth in the warmer summer months, and then during those colder months, and especially during dormancy, there's little to no growth, and so they never really reach those large reef building sizes that we see down in tropical reefs. They do secrete skeletons. They deposit these hard rock skeletons, just like the reef building corals down in the tropics, but they remain anywhere from about silver dollar size to maybe like a small coffee cup saucer in size. So they're not the huge corals that one might picture in the tropics.
And Sean, what would you add to that? What are some other distinctive features that we may not think about when we're thinking about these corals?
Oh, just how variable their colors are. I think for years we've been talking about this coral as being like brown with zooxanthellae or white without zooxanthellae. And that's a little concerning because even though they're white, they may have zooxanthellae. It's just not perceivable by us. We can't really see the amount that they have. But they can actively photosynthesize even though they look white. So I think that it's that dynamic symbiosis that deserves more study. I think it'd be really interesting to see that. I mean, when I'm diving, I see all different shades and colors and, examples of corals with and without, and some corals that have both. And yeah, it's amazing to observe.
And of course, must ask about the very important work that you are all doing. Koty, can you tell us a little bit about the Temperate Coral Research Group? When and why did you get this started?
Yes. So I would say probably back in 2009, a colleague of mine, Dr. Randy Rogen from Boston University, introduced me to this coral. I first was, when I met Randy and she talked to me about this coral, she brought it to me because I was really interested in these questions about, at the time, what are the factors that drive composition of microbiomes in corals? What controls them in tropical corals? And she said, "Have I got the coral for you?" Because I was interested in whether or not the presence of those algal symbionts could determine what kinds of microbes the coral has in it and on it. And so here we have this naturally occurring almost experimental system where we have some that are, as Sean was talking about, fairly white, some that are really brown, and then a lot that are in between. And so we can use the coral, and I was really excited to use the coral to sort of test this hypothesis that corals with different levels of algae would have very different microbes in them. It actually turned out not to be true. And then what we found was that the microbiomes on Astrangia are much more seasonally dictated. But in the meantime, what I realized was that this coral was going to be useful to so many different people who study so many different fields of coral biology. And Randy and Sean and I started this group a few years later in response to people being interested when we talked about Astrangia at an International Coral Reef Symposium meeting. What started out as a group of just over a dozen people in 2016 has now become a group of about over 150 researchers from all over the country who are studying this coral with very specific questions, and many of them are focused on fundamental biology of Astrangia, but a lot of them are also with the express purpose of applying what we learn and translating it back to timely issues in tropical coral science.
Well, I love it because I never thought I was going to be talking to a coral fandom here. So it's very exciting in of itself. And Amy, I want to ask, why is this interstate collaboration so important?
Yeah, that's a great question. I was actually just reflecting on this as Koty was talking. I think what's so important is that the questions that we're asking are still primarily foundational biology and ecological questions, which, even though there's probably thousands of people around the world studying corals, we still know very little about how they work and how they respond to stress and how they get their symbionts. And the work on Ostrangia is really focused on those foundational questions. And what I'm seeing in tropical corals now, and which includes a group here at Woods Hole that I lead called Reef Solutions, is that they are in such dire straits that we're working on applied biology towards solutions, and we're trying our darndest to use foundational knowledge to develop these solutions. But and solutions are happening in many tropical reefs labs worldwide. We're not the only ones doing this, but the challenge is we're developing solutions when we don't completely understand the system, right? And this has been happening in human medicine for years and years, right? You just need to do something, right? You just can't let people die, and so you do experiments and trials and things like that to get as close as you can. And Ostrangia is still doing really well here, and I don't know if we're ever going to have an Ostrangia restoration or rescue project, just because their role in the ecosystem isn't as critical as for tropical corals. The economic benefit just isn't quite there. And so it keeps us really focused on, how these organisms work, and we can take that knowledge and bring it back to the tropics. And so it's so incredible because there's so many universities and research institutions here in New England, as well as down the Atlantic seaboard, that may not have ready access to tropical corals. And so we can use Ostrangia to ask these questions and produce just incredibly valuable knowledge.
And Sean, final question here. We got about a minute. We'd love to just ask you, what do you hope our listeners take away from this conversation we're having today?
Well, one, that Ostrangia is the only coral in southern New England. It is their local coral, regardless where you live in this area. And we should all embrace it and just enjoy it, because I think just from this conversation with comments from Koty and Amy and Bill, it's not as Ostrangia as we think it is.
Oh, I was actually going to end it with "Don't be a stranger," because we definitely want to have this conversation again. So thank you so much for that. You've been listening to Koty Sharp, Amy April, and Sean Grace with the Temperate Coral Research Group. Thank you so much for such a punny time this morning.
Thank you.
I'm Catherine
Thanks for having
Shen.
us.
Tomorrow on Where We Live, we learn about the tenacious, carnivorous little turtle known as the diamondback terrapin. It's NautiWeek. Tune in to all of Connecticut Public's talk shows this week for nautical conversations. You can find out more at ctpublic.org/NAUTI week. Today's show is produced by Katie Pellico. Our technical producer is Kat Pastor. Download Where We Live anytime on your favorite podcast app. And thank you so much for listening.